Hi, everybody. We're going to talk about how neurons function now. So, two very important properties that we've talked about in the muscle tissue as well is that neurons are irritable or they are able to respond to a stimulus and they can conduct that impulse to other neurons or to other body tissues like muscles, glands, stuff like that. Okay. Hello. There we go. So, what makes a neuron special, or what makes it able to respond to or pick up stimuli? It's because it has a membrane potential. And so, what is a membrane potential? It is the separation of charges on the outside and inside of the membrane. So, in a neuron, the outside of the membrane has a very positive charge and the inside has a negative charge. The positive charges are caused by a large amount of sodium ions that sit very close to the plasma membrane. The inside is mainly caused by the large proteins as well as some anions that are found within the cell. And so, how did we figure this out? I mean, how would anybody figure this out? This is crazy, but they can take electrodes and insert them into a cell, and they can look at the outside and the inside of the electrode to figure out what the charge is on each side. And that difference in charges is about negative 70 millivolts, where in muscle fibers it's about negative 90, but in neurons it's negative 70 millivolts. And so we're talking about a cell membrane. In all cell membranes, we have protein channels that allow for different molecules to move through. So we have potassium channels. And so that's what these are right here. They're just showing an example of what a potassium channel looks like. We have sodium channels. Sodium channels tend to be more gated. We might have a few that are flow through, but mainly they're gated. And then we also have some gated potassium channels. So I put different colors for each of them, okay? And so remember, potassium is a positively charged ion, so is sodium. And so I mentioned that membrane potential is about negative 70 millivolts, the resting potential. There's another number that's really important in neurons, and that is the threshold. Threshold is where if you increase the amount of positive ions inside the cell and make the resting membrane potential change up to the negative 55, then you're going to have an action potential, okay? And so how does this happen? So an action potential occurs when the inside of the cell becomes more positive than it was, and then the outside becomes more negative. So if one of those protein channels opens and sodium rushes in here, that is going to make this region more positively charged. If that positive charge reaches negative 55, that's when it changes to an action potential. And so negative 55 is called threshold. Once we hit threshold, then an action potential will occur no matter what. So this is also an all or nothing mechanism. So, sorry. If and only if the membrane potential reaches negative 55 millivolts, will you have an action potential. But if it does reach 55, then you're going to have an action potential. You can't stop it. And so what happens in a membrane or in an action potential is something stimulates that plasma membrane, opening up those gated channels, and sodium channels open and sodium rushes in very quickly. So that brings a change in the membrane. So look at here, this is all positive, and now out here we have a negative charge, okay? The next step is going to be potassium channels are going to open. And so here you have that depolarization. This is from actually my OpenStax. It's a very pretty picture. Depolarization, sodium channels open. Well, potassium channels are also going to open, but they open up a little slower. So they open up and then potassium leaks out, bringing back that resting potential. So that's called repolarization. So now we go back down to that negative 70 millivolts, or we sometimes even go lower than negative 70 millivolts. And so that's where you see this repolarization. And then sometimes you even hit hyperpolarization where it goes below that negative 70 millivolts. And so at this point, I always like to ask if you can see anything wrong with this figure. So we have the positive charges on the outside, the negative charges on the inside. So this seems like you have a perfectly happy neuron cell. But out here, or in here, I should say, we have sodium. Out here we have potassium. That's the wrong configuration. If something else stimulates that neuron, sodium channels open and sodium is going to rush in. That's not going to work because there's not sodium out here. So what would happen is the sodium channels open up and sodium might try to rush out, which is going to hyperpolarize even further that cell. It's going to make it more negative. And that's not going to cause an action potential. So we have another step that has to happen, and that's the use of a sodium-potassium pump to bring about the normal configuration. Sodium has to be pumped out, potassium has to be pumped back in. These go against their gradient because remember out here there's a lot of sodium. Not all the sodium rushed in, so sodium is going to move into the cell. I'm sorry, sodium moving into the cell is going to be through facilitative diffusion, but when it pumps out, it's going to have to be using energy of ATP. Same with potassium. And so let's look at this. Here's my sodium and potassium pump. This is a sodium ion. This is a potassium ion. So right now we have potassium out here and we have sodium in here. But we want the sodium out here and the potassium in here. They're both going against their gradient, their concentration gradient, because we have more sodium out here than we have inside, and we have more potassium inside than we have outside. So 3 sodium can bind to the sodium-potassium pump at its normal or in its resting configuration. When ATP binds, though, it activates the pump. So here's your ATP, it binds, and that's going to change the shape of the pump. So the changing shape of the pump allows sodium to be released because it can no longer bind, but now the new shape lets potassium come in and bind. The ATP was hydrolyzed to activate the pump, and so when the phosphate is released from this, it's going to go back to its resting configuration. So there you go. And I'm sorry, this is my animation. It's a little slow. And the potassium is released to the intracellular region. And so now more sodium can bind in its resting configuration. So here are all the stages of the action potential. So you're at this resting potential. Some sodium might fluctuate and come in, and some potassium might go out, and that leads to these graded potentials that could occur. But then if enough sodium rushes in that it reaches that negative 55 millivolts, then you have an action potential. And so that's going to increase the positive charge inside the cell to about positive 30 millivolts. Then sodium channels close, potassium channels are opening, and potassium starts rushing out. That brings us back to resting membrane potential, and sometimes we go a little further to a hyperpolarization. And then we go back to rest, okay? So let's look at your version. We start with this resting membrane is polarized, so it has a difference in charge. Outside is charged by the positive ions, inside is charged by the proteins and the negative ions, but we do have a lot of sodium outside and we have a lot of potassium inside. A stimulus hits, and when it hits, it allows sodium channels to open. So any stimulus causes those gated channels to open. Sodium rushes in. If enough sodium rushes in that it brings the membrane to about positive 30 or negative 55 millivolts, then an action potential will occur. So depolarization is going to, so if you hit that negative 55, you go into the first major step of the action potential, which is that depolarization step. And so depolarization, lots of sodium floods the inside of the cell, and that makes the inside much more positive to about positive 30 millivolts. This also leads to threshold in the next section. So this area here is going to hit threshold, and you're going to have the action potential move. So remember that propagation of the action potential, this is how it occurs. So now we have the next section becomes more positively charged, and an action potential hits there. As this is occurring, actually the ones back here, this region is going to go into the repolarization mode. It's almost like when you're doing the wave, like if you're in a long line, one person stands up, puts their arms up, and sits down. The next person's already standing up. And that's what's kind of happening. Think of it that way. So you have one person at a time standing up doing the wave, and each person is a section of that plasma membrane. And so the first region would be where the depolarization started, and then the next region would have depolarization while the first region would repolarize. If that makes sense. So as one area is, as the action potential is moving in one direction, behind it you have repolarization. So potassium channels open and potassium rushes out of the cell, bringing back that resting membrane potential. So now you have potassium outside and sodium inside. Sodium potassium pumps bring back normal configuration by allowing sodium to be pumped out and potassium to be pumped back in against their concentration gradients. This is an ATP pump, so it takes energy. At this point, you should be able to tell me or answer the questions. How can you classify neurons functionally and structurally? What is a resting membrane potential? What is an action potential? And what occurs during an action potential? I'm going to stop here, and then we'll get into transmission of nerve impulses at the synapses, okay?